Good morning, everyone. Welcome to our fall member event. I'm thrilled to see such a great turnout. I'm Kris Rebillot. I'm the Director of Communications here at the Buck, so I'm going to be moderating and kind of hosting today's event. First logistic question, if you can, please turn your cell phones off, or at least turn them to mute so they don't interrupt everything. Plus, our sound system works better if you have your cell phones turned off or turned down. Before we get into today's presentation, I want to give you an update on some of the research that's been happening here at the Buck Institute, especially in the last couple months. So, first up, from the Verdon Lab. This research got a really big splash. See, there was an article in Wired. A ketogenic diet protects memory in aging mice. So that's a high-fat, very low carbohydrate, low protein diet. You may hear more about that today because that research in ketogenic diets is applicable here at the Buck to more than just memory. And then breast cancer, developing new prevention strategies for breast cancer. That's from the Benz lab. And what the researchers discovered, it's kind of dense, but I think it's really interesting. They found that women who had high blood pressure during pregnancy have up to a 90% reduced risk for breast cancer if they carry a very common gene variant. So what they're trying to do now is they're delving down into the molecular pathways involved in that genetic protection, and they hope to exploit that for the benefit of all women. So it's a new way of looking at preventing breast cancer. The work was an outgrowth of the Marin Women's Study, if you remember. That was started in part here at the Buck Institute many years ago, and then involved the California Teachers Study, which involved more than 133,000 women. So we look forward to more from the Benz lab on that. And then we have a new scientist here at the Buck. We're very excited to have Dr. Kai Zhao on board. He came to the Buck with a $2.4 million grant from the NIH. It acknowledged him as an exceptional young scientist, which means he's so good he can skip his postdoctoral training and he's going directly to do his own research. Kai is exploring a very big idea in aging research. He studies proteins folding and misfolding, and his idea is to hone down on the cellular level and to try to figure out a way to turn back the clock on a cellular level, so to reverse the aging process. So we welcome Kai here. I don't know if you noticed when you were out in the atrium this morning, we have new trees out in the atrium. Our fruiting olive trees had lived a very long, healthy lifespan for 20 years. It was time to have them replaced. The new trees out there are called Ficus benjamina. They're native, they're the national tree of Thailand. The trees that are out there came to us from Florida. Through San Diego. It took a long time to get them because the hurricanes in Florida wiped out the ficus nurseries, but we finally got them. And supposedly the highest tree that we have is 15 feet. We're told that they can hopefully grow another 20 feet. So we hope to have even bigger trees in the future. So, final question before we move on. How many of you here today are guests? All right, we love having guests. I'm going to make a pitch for you to please consider becoming a member after today's event. We have a sign-up sheet, a sign-up person outside that you can talk to. I think I love having members. I always say that our members are some of the smartest people in Marin County because they really do keep up on what's happening here at the Buck. With the holidays approaching, I want to suggest too that if you have friends or family and you don't quite know what to get them for the holidays, give them a gift. of membership at the Buck Institute. So that's my pitch for you to join and help in our research. So next up, I've asked Dr. Gordon Lithgow, who's a longtime faculty member here, he's our Chief Academic Officer, to introduce today's speaker and to put today's presentation into context. Gordon runs a lab that's focused on increasing lifespan and health span by enhancing molecular stability. He's also one of our most popular speakers. I think those of you who are members have seen him before. So, Gordon, it's up to you. Take it away.
Thanks, Kris. I'm very honored to be introducing Pankaj to you today. Pankaj has made many incredible discoveries here at the Buck, and I'm going to take 3 or 4 minutes just to talk about that process, because it's something that I just want you to be aware of how special this is. And to do that, I want to put you in the shoes of a scientist. So imagine that today is your first day at the Buck Institute, you've scored a job here, and you find yourself going into a lab, much like this one, which is down below in the floor at the back there. And you're assigned to a bit of bench, about 1.5 yards of bench, where you'll keep some small equipment, you'll keep your lab book, and this is where you're going to be spending many, many years doing research. And what do you bring to this? Well, you're going to have to rely on your training. You've probably got a PhD in molecular cellular biology, and you've read the literature, and you're aware of some of the big questions in aging and age-related disease. But pretty quickly, you're going to have to come up with an idea, a new idea, and an experiment. And this experiment is going to be something that no one else in the entire history of the world has ever done before. No pressure. So what do you need? Well, you'll probably work on either worms or flies or yeast or human cells perhaps, neurons on the right and fibroblasts on the left, or you might be working with mouse models. And you'll need equipment. You'll need glassware, sterilized glassware produced by the Institute for you to use and get started with your experiments. You might need some really expensive equipment like this one. This is a protein mass spectrometer, about $1 million. Now you're new, and so you're not going to get to touch that equipment. You're going to have to go talk to Birgit Schilling, the head of the core. And Birgit's going to be skeptical about your idea, and you're going to have to sit down and talk to Birgit for hours and convince her that it's a good idea for her to run your samples on her very expensive machine. So you do that, and you might even need one of those, a white coat. So you go back to your bench, and it might take a few weeks, unlikely, it's probably going to be more like 6 months or a year or 2 years before you make a discovery. But when it happens, it's instantaneous. You're looking at some flies in a test tube, maybe you're looking at some cells down a microscope, and it's instantaneous. You immediately realize you're seeing something that's new, that no one else in the world has seen before, and you've made a discovery. And that's a very special time, because you are the only person in the world that knows something about life, about biology, about a disease. You're the only person in the world that knows it. No one's ever seen this before. And you can choose to share that right away. You can turn around and say, hey, do you see this? Am I seeing this the way you're seeing this? Or you might choose to keep it to yourself for a few hours or a day. Special 24 hours in your life where you know something about life that nobody else in the entire world knows. But eventually you'll start talking to your colleagues, and you'll need to convince them that this observation is real, and you'll do more experiments. You'll do more and more, and you'll eventually get to a point where you have to convince a whole lot of other people. You'll go to a conference and speak in front of 100 skeptical people about your idea. But eventually you'll write up this idea in a paper, in a scientific journal, and you'll send it off to the journal, and you'll hear back from the journal. And you'll only then realize that there's a whole bunch of other people who are skeptical, and they want you to test your idea in 8 ways that you never even thought of before. And so you go back to your little bit of lab space, and you do more experiments, month after month, year after year. And usually 1 or 2 years after you make that original observation, that special moment that happens instantaneously, eventually your science paper comes out. And it comes out into a cacophony of other science papers and other discoveries that people are seeking attention for. So that's the process. It's a very special process. It's very challenging. It takes a lot of time. It's a team effort. You've got the whole institute behind you, pushing you towards that special discovery. But it's very joyful as well. And Pankaj is a special scientist, and just based on his publication record, I think he's done this over 70 times. That journey, over 70 times. It's really remarkable. So To introduce Pankaj, Pankaj obtained his PhD at the University of Manchester in England in the biological gerontology unit there. I was fortunate enough to share an office with him for 6 months during that time. I learned that he's an incredibly creative thinker. He's a very deep thinker, but the thing that's really special is his passion. He was an incredibly passionate PhD student, and he brings that passion to the Buck Institute every single day. He went to work at the University of California, San Diego, before going to Caltech and working with Seymour Benzer, perhaps the greatest scientist of the 20th century, at least some of us believe that. Seymour made incredible discoveries, including defining what the gene was, and then went on to work on areas that Pankaj will probably talk about today. So I'm deeply honored to introduce Pankaj. Consider him a great colleague and a great friend. So. I'm Kris.
So here's how it's going to work today. I'm going to be your stand-in initially. I'm going to have a conversation with Pankaj about his research. I think you were given white cards to write questions on. If you have a question, raise your hand. I'll have a volunteer come and pick it up and bring it to me. We found with the cards, it's really easy for me to identify the most popular question that people have. So that's that. I also want to acknowledge Pankaj for being willing to let me hold the slide clicker. Believe me, for a scientist, that's a big step in giving up control. So I have the clicker. All right. So welcome, Pankaj.
Thank you.
So before we get into a lot of the projects in your lab, and I want to say that Pankaj's lab focuses on the big question, as we say, nutrition, metabolism, and aging. We're going to talk about a lot of his projects today. His work has a direct impact on health in terms of diabetes, obesity, kidney diseases, circadian rhythms. We're going to hear about that today. So there's intestinal permeability, leaky gut. So there's lots of connections, and I hope to be able to get to all of them today. But the first thing, let's talk, Pankaj, about human beings. So what happens with our metabolism as we age? What generally happens?
Well, things fall apart, unfortunately. But what we're realizing is that so many systems in the cell fall apart, but metabolism is very central. I mean, basically our ability to generate ATP falls with age. So there have been very nice studies showing the ability to make energy for the cell is just declining with time. And we use flies and worms models to really understand, is the same thing happening in a shorter time frame in these models, and can we understand something about this very complex problem in humans which takes decades to build up? Can you model that in an organism that lives only about 2 months or 2 weeks, and can you study that? Yeah.
Perfect segue. So your lab works primarily in flies and worms. Why should we care? I remember we were talking this morning when Sarah Palin ran for vice president. She slammed research that happens in fruit flies. We think it's pretty cool. Why is it, Pankaj?
Yeah, I mean, I want to thank Gordon for the introduction. It just reminded me of the conversation I had with Seymour Benzer. So Seymour Benzer was one of the first people, one of the pioneers who adopted the fly as a model system to study behavior, circadian clocks. Actually, the last year's Nobel Prize was given for circadian clocks, and it was work that started in the fly. So I remember him talking when I read his biography, and he was simply interested in why his 2 daughters had different behavior, and he just got very interested in this question of what dictates behavior. In 1970s, people thought it was a very amorphous concept. People thought we have no clue, and definitely no gene had been ascribed to a behavior. And at that time, people also realized we had clocks, plant had clocks, right? But nobody really understood why and how this whole thing works. So his work really pioneered this field. He found the first gene that dictated circadian clocks. And what I mean is it's a very, genetics is a very beautiful process. So he found that flies had certain behaviors that take place at certain times of the day. So for example, they lay eggs early in the morning. So they said, okay, why do they lay eggs at a particular time? Then you do something what's called mutation analysis. So you just find strains of flies which are different, which don't behave just like us, right? They found strains of flies that would lay eggs all throughout the day, right? That very simple discovery led to the identification of the whole molecular pathway for circadian clocks. So,
and that was involved in the Nobel Prize this year, right?
Right. So unfortunately, Seymour passed away, but he is credited with starting this whole field. But because if you pass away, you don't get the Nobel Prize. So his people who carried his work on, they got the Nobel Prize. But I talked to him, and I remember talking to him on the phone. And it's like you asked me, what happens in humans? I was like, I'm interested in human aging and human diseases. What am I going to learn from the fly? And he's like, no, that's what we're studying. We're studying human diseases in the fly. And it just made a very strong impact on me when I worked in his lab, that you can actually understand so much about a biological process from these simple systems that would take you years, that basically would take you far too long in other systems. Not just because they live short, just the way you can manipulate them. I sometimes joke, we can play God with the fly because we can change any gene at any time and look at their behaviors in a way that would not be possible, right? And where I'm trying to do work in mice and humans, and the number of things you have to sign to just something simple is just incredible. So it's just a very difficult process. So it's been a gift, I feel, to me, and changed my career trajectory to work with Seymour, and just made me realize. So now it's more imagination is the limit. So what do you want to understand? And I often ask this question to the audience. Come up with a question, and we can see if we can model that in the fly. And like that, we've at the Buck been privileged to be able to do model a lot of interesting things in the fly relevant to aging and nutrition. Yeah.
That's great. So a lot of the work in your lab involves a pathway called TOR. I promise you this is one of the most complex slides today, but I'm committed to having Pankaj explain it to us in a way that we can all understand. So talk about the TOR pathway and why that's so important to your work.
Could I have the pointer?
No, you cannot.
So the idea is that TOR has been discovered as a link between nutrients and growth, which means every time we eat, how does think about an organism, it eats and then it grows. How does that happen? So there's a process in place which is dictated by a protein called TOR, which is key in that if this protein is not there, food does not turn into bigger size. So this is very simple but very important discoveries. What's amazing is it's the same protein that does this from plants to humans. That I find just amazing. And it sort of talks about the common origins of evolution, common ancestor, but it makes sense why reinvent the same thing again and again. TOR does this job beautifully. So what it does many things downstream. There are many labs around the world trying to understand what TOR does, but simplistically, it's what we call a nutrient sensor. So as soon as you eat, this is getting activated and it's turning and putting the animal in what we call the reproductive mode or a growth state. And when I was a postdoc in Seymour's lab, I thought, okay, if this is true, then I knew something else from my prior background, which was that, well, if you eat less, you can live long. But till that point, it hadn't been done genetically. You had to really eat less to get this benefit. So people didn't understand why you're getting all this benefit. So I asked this very simple question using the fly. Well, what if we take out this protein or turn it down so that, every time it gets food, it doesn't start, go into this drive to grow. So question is, we ask the question, if you turn down TOR, can you put the animal in survival mode? And what I mean is, when you eat less, the idea is you live longer because you get into this survival mode, and imagine in the wild, right? If you eat less, what are you going to do? You're really saying, okay, we've got to conserve energy. I got to somehow maintain my body till I find food, and then I'll be able to reproduce, right? So all organisms, that's why bacteria form spores, and all the species have some mechanism in place to be able to do well when under difficult conditions, right? So starvation is one of those conditions. So that's the survival mode. So what's cool about that mode is you get a sort of byproduct of that is that you live longer. So if you can trick your body into being in that survival mode, your body is waiting for this food essentially, but you can trick it to live longer, right? And that's what's happening in the wild. So that's what we found when we turned down TOR. And not only did it live longer, it's now been linked to increased slowing neurodegeneration, and it was a big target for cancer already. It was known that if you turn down TOR, you would reduce your risk of cancer, which is, as you can see, it's tied in with growth. More TOR is going to be more growth of a cell. So it reduces risk of cancer, reduces risk of age-related diseases, and extended lifespan. And that, I just want to say, that work then got reproduced in, I think, over 50 studies from yeast, like I said, to plants, from yeast to worms to we did it in worms and flies. Now it's been done in mice, and now people are even finding same thing to be true in humans. So this seems to be really conserved across species. Yeah.
So what about this? So we're talking about food scarcity, but food abundance. And I know a lot of your work focuses on problems that happen with overnutrition, right?
Yeah. Well, there are two sides of the same coin. On one hand, we can ask what are the benefits of eating less, but on the other hand, we're facing this epidemic of obesity. So we're asking, well, what happens if you go into this state of overdrive and overnutrition? I find this slide very important. The reason is, everything we do right now, we have to remember, we are in this snapshot of time right here. It's been hundreds of millions of years of evolution that's brought us to this point. And the reason that is very important is as we start looking at the genes and we try to start studying the lab, there's a very important quote. Oh yeah, it's there. Nothing in biology makes sense. I would say actually nothing in life makes sense except in the light of evolution. So because I think it applies to relationships, it applies to marriage, it applies to everything. But especially in regards to what I'm talking about, aging, and what has happened is there are many processes that have evolved that were important when we lived under relative food scarcity. And now, when we're suddenly faced with all this excessive food around, we really don't have the control or the mechanism to deal with it, right? Because we didn't evolve as lab rats, right? So the way, for example, people blame fat. Generally, people think fat is a bad thing. I mean, absolutely not. Fat is a protective tissue. Without fat, we wouldn't have been able to survive the boom and bust economy which every organism faces in the wild, right? Fat was there to make sure that when resources are, like I said, diet restriction, when resources are not around, that you can survive for longer periods of time. Now, problem is, when you are overeating, that same fat turns against you. So for example, I don't know if you know, but people who are diabetics, I mean, one idea is they're diabetics now because they're overeating, but if they were actually under conditions of, if there was famine, they would actually do better than the normal people because they would have better stores and better ways to deal with the lack of food. And that's the problem. So it's a trade-off, and this is happening again and again in many situations. We see this. So I just wanted to remind people when they're thinking of this, you can't forget the picture. Yeah.
So now we're going to talk about some specific projects that are happening in the lab. First up, let's talk about circadian rhythms. I know most people think about circadian rhythms in relationship to sleep, but it's more than that, right?
Absolutely, yeah. So it was a education to me as well. I'll tell you, one sad story is that when I was in Seymour's lab, I knew about circadian clocks, but I didn't put it together with aging. It took me a while when I left his lab that, this is, now I know you guys are going to say, yeah, I agree, I'm not smart. It took me a while to get this together. But basically, the focus had been on this circadian clocks regulating sleep and behavior. But what became apparent, and that's big reason why they gave the Nobel Prize, big reason, and they're realizing now, it's tied in with metabolism, aging, and so many diseases. So animals that don't have a proper circadian rhythm get obese, get type 2 diabetes, get cancer. So they're realizing that it's tied in with too many processes. And the key concept to remember is that it's not, like I said, it's not just sleep. Every cell in your body has a clock. And that really getting into this project changed the way I'm living a little bit, because earlier I remember I wouldn't think twice about going to the refrigerator at nighttime and I'm hungry, I want to eat something. Never thought that was a problem. Nobody knows if that was a problem, right? Till recently they've done studies and basically it shows that if you eat at nighttime, that really predisposes you to obesity. And if you were to not eat at the wrong time, basically doing things at the wrong time, it messes the body up. The idea is that, like I said, every cell has a clock. So we're tuned. The body is tuned in. So the liver and the gut. Think about that. They wake up in the morning and they're ready to take in food and digest it and process it in a certain way. So there are processes for the day and the processes for the night. So during fat, for example, you've got to take in the food, assimilate it as calories, and then at nighttime when you're not eating, you've got to be able to break down. There's partitioning. All these processes are partitioned. Now what happens when you eat at the wrong time? The body gets the wrong signal. It's like the gut, the liver, it starts waking up, and you end up actually, this has been done in mice. They took two populations of mice, one that fed at night, one fed at day. Mice wake up at night. The ones that eat at night are totally healthy. Same amount of calories. If they eat during the day, they become obese.
And mice are on a different schedule.
But they're nocturnal animals, right. So I hope I didn't confuse anyone. So the idea is that the other thing we're finding, which is very exciting, is we're finding that a lot of the repair processes take place at night. So think about the consequences, again, of giving food at the wrong time or not being able to sleep. You're not giving your body a chance to essentially regenerate. A lot of stem cell, you hear a lot about stem cells. Well, all that's happening at nighttime. Well, a lot of the regeneration, repair takes place, DNA repair, dealing with oxidative stress, all that, the body is trying to fix that at night. Food created, for example, food and other things you did during the day created a lot of damage, and that's getting fixed. And if you don't do that, we're finding lots of problems take place in the flies, for example.
So for you personally, how do you manage this?
Yeah, like I said, so I try to at least have, the recommendation is at least 12 to 14 hours of break from food, essentially. So when you have your dinner to the next meal, try to keep that break as long as you can. I mean, 12 to 14 hours, like I said, would be ideal, yeah. And just having improved physical activity during the day, that really helps you sleep at night. I would add to that there's more and more evidence that a lot of the neurodegenerative diseases, people are finding evidence that sleeping disorders precede the actual problems. You can have problems in your early 30s, 40s, which eventually, they think those are early sensitive markers that predispose you to neurodegeneration. So activity, and the other thing is just light exposure. We're doing this at this time. Like I said, remember that slide. This is not how we were brought up, not to be in a room at 10 o'clock in the morning. What is very interesting, and we're finding this in the fly as well, I think it's just fascinating. The body is most receptive, and the fly, most receptive around 9 to 12 o'clock in the morning to light. So if that's when you want to get your sunlight, and that helps the circadian clock the most. So we can model that in the fly. We're seeing that there are changes in the eye of the fly at that time that make it receptive to light. And if you miss that period, and so now people do that as phototherapy for even for Alzheimer's. There's one of the articles, a few articles I read, but they're doing phototherapy, and there's an optimal time for that. So, it's linked with lots of things, lots of behaviors.
So get up in the morning, open the windows, get your sunlight early, and try not
Right.
to sit in dark rooms
Right,
too
right.
often.
And be active during the day. Nothing like everybody knows here. Yeah, you swim. The day you swim or you do something, you exercise well, that's the day you have the best sleep. Yeah.
So next slide. So your lab is also focused in a big way on diabetic complications. Talk about those.
Yeah, I want to, before we get into that, I want to say, like, it started with these models, this C. elegans model. So diabetic complications is a huge problem in the sense that, let me give you a little background on diabetes. So you all know of diabetes, you go to the doctor, you all know if it is a disease of hyperglycemia, your glucose is high. Well, let me give you the good news. High glucose isn't what kills you, isn't what's damaging. But the bad news is something happens after decades of this metabolic disorder, right? This decades of having high glucose, you build up what are called advanced glycation end products. We'll get into more. That's when you start getting what are called diabetic complications. You get all these pathologies. So the problem is it's a really huge problem because like half the people who have diabetes have some sort of neuropathy. You get these tingling nerves, you're sensitive to touch. It's one of the leading causes of blindness, diabetic retinopathy. Kidney disorder is one of the biggest problems with chronic kidney disease. Diabetes is one of the biggest risk factors, and people who have this, like literally, I think 20 to 30% of the people have some sort of nephropathy, which means they'll have to have eventually a kidney transplant. It's a big risk factor for cardiovascular disease and stroke. And then the other thing, being at this institute, I've been very mindful of, it's not considered a complication, but it is, because having diabetes increases the risk by almost 5 to 10-fold of getting some sort of neurodegenerative disorder, like Alzheimer's and Parkinson's. So understanding this, like I said, it takes decades to follow this up. How do you study something this complex? And I was surprised. I've talked to many clinicians. This is actually a disease of aging. Maybe more, the diabetics are more at risk, but the rest of the population is also at risk. So you get neuropathy even in the rest of the population. So, and it's tied in, like I said, with more glucose, essentially, which everybody is consuming glucose, right? So what was very exciting is we found, like I said, it takes decades to build up, so not many people study this. Clinicians leave it alone because they say, well, this is just fait accompli. This is just going to happen to you. We're just going to focus on treating diabetes. But what we found was a very cool model in C. elegans. We were able to see this in a matter of 3 weeks. And that changed the way we've been doing things in the lab.
All right, so now we're going to talk about, hope that'll come up.
So, right, advanced, I'll just go step by step.
Okay.
So, like I said, glucose elevation is a problem in diabetes, but we think that's not what kills you. What causes the pathology is something way more reactive that is produced downstream. This is, I'm sorry for all the formulas and chemicals, but that is a much more reactive chemical. We call it methyl glyoxal. But the other chemical you've heard of, it's called advanced glycation end product. This reacts with the proteins and the DNA, and like you heard about protein misfolding. This is methyl glycol is going to modify the amino acids, and it's going to form what's called advanced glycation end product, because glycation is, over time, the glycation ends up as something irreversible. You can't change this. It builds up in our cells, and essentially that's what's causing tissue and cellular damage.
So just to be clear, because I've done the news releases for him, so I kind of understand this stuff, is that ages are formed when sugar binds with proteins and lipids, right? And it's like a toxic molecule.
Right. So I don't know how many of you think this is a foreign concept. You deal with this every day. Every time you're cooking, right? The caramelizing onions, that's essentially forming advanced glycation end products. You see that browning, you see? So unfortunately, we also take ages from our diet. So not only are we making this slowly in our body, there are mechanisms in place to deal with it, but it's happening on every time you're cooking and you're frying, especially when you use dry heat. You see how it gets dark very quickly, right? And so it's also changed my way of living a little bit. Less barbecue, definitely, but also try to steam or boil stuff, because that produces less advanced glycation end products. And what's very exciting is, exciting and sad, but we've been able to, like I said, model this in the worms, but we can feed these ages directly to the animals. So this is very important, because like I said, we're consuming these ages every time. I looked, when I got into the project, I looked up on Amazon. They sell something called liquid smoke. I think a lot of people here know that. Well, liquid smoke is essentially like eating ages. So the problem is, these ages, when we feed them to the animals, they cause all sorts of problems. They cause obesity, metabolic disturbances, neurodegeneration, shortening of lifespan. I think we'll have slides on that. But so we basically, it's never been modeled, like I said. So people have understood this chemistry is 100 years old. This part is 100 years old, but we're trying to say this is causal to disease. That link has not been established. People think this is something that just happens slowly with aging. And this is the problem. This is why I think the buck, I'm working on the buck is special. What we're tackling is something that most of medicine has neglected or hasn't had time to deal with. Which is a very slow process of aging, and how does that relate to medicine? Like I said, we're trying to understand how aging of diabetics takes place, and also that's how that is relevant to the normal aging as well. Ages build up, not just in diabetics, it builds up almost linearly in all of us.
So when we're younger, we're better capable of clearing them out?
Right, so there are enzymes in place to deal with this methylglyoxal, yeah.
And talk about, you've told me stories, the worms and the flies, they like ages, right?
Oh yeah, that's very interesting. Like I said, liquid smoke is sold because we like this stuff, right? We use it as a condiment, or we like to barbecue our food. Well, there's another surprising thing, but it tells you about the sort of the deep, sort of conservation of these processes across species. Our worms, which are these 1 millimeter long animals and flies, they also like the taste of this stuff. Yeah.
Makes you look at French fries differently, unfortunately. So you mentioned it briefly. How have you changed how you eat based on what you've seen in your lab?
Yeah, not very popular in parties now. I always yeah.
So you talk about not using as much dry heat, though. So how do you try to cook?
So when you can use steam or boiling, yeah.
Okay, all right. There's another.
Or eat raw food. You can. Yeah, no need to unnecessarily cook stuff. Yeah.
So one of the projects that you do, we don't have a slide for this, but you have a project with the Verdun Lab involving a ketogenic diet, which I talked about earlier. Are you looking at ketogenic diets in relationship to ages? Talk about that.
Yeah. So the idea is this is a byproduct of what we call glycolysis, of burning sugar in the body. So what we're finding is, if the body is in the sugar burning state, then you produce more ages. And if you're more in the fat burning state, you're going to be better off. So what puts you in a fat burning state? So Eric, they published one way is to just eat less of the sugar. That definitely works, by the way, to some degree. But there are many other ways we can do this as well. So, for example, every time you exercise, what happens is you run out of glucose very soon, and the body has to start using other sources of energy. It's fat. So the key thing to understand is that you want to put your body in the state where it's learning to burn fat. And that's what happens when you're exercising for longer periods of time, especially. So you don't want to just do 5-10 minutes. A fit works, but the idea is that the prolonged exercise. And one of the things I used to do was eat before I exercise. After talking to John, I realized no, we want to put ourselves in this state of burning fat. So you want to eat before, hours before, and not right before, and get the sugar load and try to avoid the starvation you get after exercise. But what's interesting is the body learns, and this is very important because the body learns and it can deal with these, keep us in a state of burning fat for longer. The other thing that's very important is that just the type of diet you're eating. We're finding the sugar burning state is also a sort of sugar addiction. You go towards more carb-rich food, it is not as satiating to the body. So you need more. And it becomes a vicious cycle. You're burning sugars, we're producing more ages, we're finding ages actually make you eat more. So you get into this vicious cycle of trying to eat more and not really being, always being hungry, versus if you were to more go to like a nuts-based diet, which is, the idea that would be more ketogenic diet. There are many good ways to do, I guess, ketogenic diet. But that would, essentially lowering carbs will put you in that state where it's learning to teaching the body to burn fat rather than be reliant on sugar all the time. Yeah.
And
it's very hard to tell this to my daughter, but I have to do this every day. Every day is in our household we have to discuss this.
So in your lab, you're working on compounds that deal with ages, right?
Right. That's so we've got a whole project around ages and understanding this at many steps. Many of the genes that are involved in regulating this pathway are important disease-related genes, important for cardiomyopathy, all the complications I talked about. If you have genes that predispose you to make more ages, we're finding that might predispose you to have those diabetic complications. So we've got compounds now, and actually turned out some very simple supplements as well that can keep these ages at bay. So we're getting really amazing results in these animal models. And one of the things I wanted to say is I started in medical school. I was very interested, like I said, in human disease, but I never thought I'll be getting into clinical trials. But some of the work we're doing with this in worms, finding new pathways and compounds, we're now looking for clinical partners. We have a friend at now UCSF who's going to do a study with these supplements to see diabetics if this would reduce weight gain and glucose in diabetics. We're trying to do another study with a group at UC Davis to see if it affects chronic kidney disease. So I'm very interested in trying to reach out and see where this is relevant. And we're working with many people at the Buck, for example, Julie Anderson, to see its link with Parkinson's. So it turns out one of the genes that causes predisposition to Parkinson's is directly linked with making more ages. So we're finding another genetic link, very, again, strong genetic link that says this might also be relevant to Parkinson's and Alzheimer's disease.
So I know you did one study in your lab that involved a supplement called alpha-lipoic acid, and that's I think it's okayed in Germany, right, for diabetic complications? So talk about, because that's something that's available now.
Right, is that on the next slide?
No, we don't have.
Okay. So what we discovered, so alpha-lipoic acid, again, common supplement, has been given for a long time, and it was approved in Germany and Japan, but people they found different effects with it, including lowering of glucose. One of the things we discovered is that it lowers advanced glycation end products. What it does is it switches on the enzymes that detoxify methyl glyoxal, the damaging product. So by that, it's lowering the ages. And like I said, I take it now after discovering that. So I want to say this. I think I skipped that part. The way we discovered this is in C. elegans, right? This little worms that Gordon also works on. Remember, I talked about neuropathy. In neuropathy, people become sensitive to touch. There are special cradles sold, in shops, just so that you can rest your feet, because even the touch of bed sheet is actually going to be very hurtful. That's what happens when you have neuropathy that gets early neuropathy. Eventually it leads sometimes to amputation because you can't deal with infections or other problems in like the extremities, right? So it turns out our worm model, when you have more ages, also becomes sensitive to touch. So we think it's remarkable. It's like same way the human neurons become more responsive to touch, we're finding that in the worm. So using that model, we discovered that this commonly used drug, alpha-lipoic acid, might be very effective. It is effective in the worm and might be effective in some people. Yeah.
That's great. Okay, so then let's talk about. It's not doing it. Let's see.
That's to avoid.
Yeah,
My
right.
pointer
right.
to remind everybody to be unpopular.
Okay, so you're also in your lab working on something that's called intestinal permeability, more popularly known as leaky gut. So talk about what is leaky gut and why is it a problem?
Yeah, so one of the things, like I said, one of the advantages working with these model system flies and worms is you end up discovering new things. So what happened is one of my postdocs discovered this, so we've been working on how, rich nutrients shorten lifespan, but, there's so many things happening. He came up to me one day and said, I think these guys have leaky guts. And we say, how? How do you know? So he says, well, I fed the animal this blue dye, and the whole animal's turned blue. Because if the gut is leaky, and we can do this sort of experiment in the fly without feeling bad. So we get this blue fly, and we call this assay a Smurf assay because the whole animal looks like a Smurf. So then it turned out, led us to a very interesting discovery. We found that imagine all your epithelial cells, all your cell layers that are protecting you from the environment. So the gut layer is one of them. So what's happening is there's a breach there, it's not getting repaired. So it led us to a very interesting discovery that these cell layers, imagine a cell layer, the cells have the job to respond to, take in all the food, digest all the stuff, but all the damaging stuff that's happening, it has to deal with that. And what happens if the cell is faulty, right? What happens is when the cell is faulty, we found that the neighboring cells get rid of it. There's a quality control mechanism. It says, this is not a fit cell. And same thing's happening in your skin all the time, otherwise there'd be huge holes here, right? And the gut, is the tissue that turns over the fastest in the body. 3 days, we almost get a new lining, right? So, but there is a very exquisite process trying to get rid of bad cells. We find that as you age, that's not as efficient. That's what we were able to find. And if you eat a very rich diet, the flies were very inefficient at repair of this sort of damaged cells and sort of sealing the layer. So under dietary restriction, you get this optimal repair. You get the cells, the bad cells taken out. Under rich nutrient conditions, they just stick there, and the gut becomes leaky.
And so then toxins get into the bloodstream?
The toxins get in the blood, exactly, and that's well described, and we've been able to redo the whole study in mice. Same thing happened. Put them on a high-fat diet, not ketogenic, by the way. This is high-fat and high-sugar, bad diet. The guts become leaky, and they all the bacterial. So essentially, what happens is your microbiome, which is very useful, very good for you. We have a very symbiotic relationship with the microbiome. But if you have a leaky gut, then it wreaks havoc because the bacteria are just going to go through and do what they're not supposed to do.
So there's a lot of articles in the popular press. Everybody's really excited about the microbiome and all the flora and good bacteria.
Right.
Do you take probiotics? And what's the takeaway for people here? Like what?
I have to say there's some evidence, and it can't harm you. Have yogurt. I've switched over to yogurt from milk whenever I can, because there's a lot of bacteria we've again grown with. But I do take probiotics because I think the good bacteria can sometimes even help in this repair. But like I said, if this is faulty, then nothing. Then you're in trouble. Yeah.
Okay, great.
And it's been linked with neurodegeneration because, like I said, imagine this stuff going into our serum. Well, now it's going to increased risk of cardiovascular disease, and because it's going to travel to other spaces where it's not supposed to be, it can increase the risk of neurodegeneration as well.
Okay, great.
And so I didn't believe this. Like, how the hell can gut be tied with it? But we've done these experiments in the lab. We've created animals with the leaky gut, and we have a model. We look at their brain degeneration, and yes, they suffer more damage to their brains. Yeah.
All right.
So that we can have cause and effect. That's one of the most satisfying things working on the worm side. We can talk in terms of cause and effect. We don't, if there's an association, we test it. We don't leave it at that. Yeah.
So if you have questions, now is the time to have them passed up to me. We'll have a volunteer pick them up. So this slide will make sense in a minute. So let's talk, Pankaj, overall. Do you think that there is a particular diet that's good for aging? How should individuals look at
right.
diet and aging?
So So I have to say, when I started my lab, it was with the naive idea, diet restriction works. I have to say, this I have to share with you guys. When I think I was 19, 20, when I learned about diet restriction, I was so excited. I was like, I can't believe there's an intervention out there that gives you an extra decade or two in life, and you don't have to do anything. Well, you have to eat less, but you could really get this, and you get a healthy life. So I've been very fascinated with that, even though I'm very drawn to food, but it keeps me away, just this knowledge, right? So, and the naive idea was that I think it's going to work in all. It's been shown to work in 30, 40 species, and it should work for everybody, right? Well, so there have been some reports saying, well, it works not the same way for everybody. So like I said, we don't leave things to association. We tested in the lab. So we had a very nice collection of fly strains obtained from the wild. So now we're not working on one strain. So remember, all of you, we're all different. So this project is all about individual differences. So we got 180 fly strains from the wild. All of them are sequenced, which means we know exactly the DNA of all of them, right? We asked the question, how do they all respond to diet restriction? So the idea, another way, if I was to ask all of you to go back and do diet restriction and then took measures on all of you, what would it look like, right? And that's the experiment. That's the big experiment, right? So we did this in the fly. And the reason it's important to do this in here, because you learn so much here that you could apply this knowledge before we do these crazy things in humans, or we could have very much more, we'll be better informed, but we won't be surprised. So for example, they did a primate study, started 35 years ago, and they said, well, it doesn't work, and it makes big news. Well, there were many things that were not done right in that study because they didn't really quite know 35 years ago how to really do a study for caloric restriction, right? So I'm just saying, so you only learn, it's easier, hindsight is 20/20, but the idea is that once you do experiments like this, you learn something. So what you see is on each of these lines represent the mean lifespan. That's how long the flies live. Some flies live only about 20 days, so the mean lifespan, let's say, is about 10 days, right? So in red is the lifespan of all these flies, of all these 180 strains on a rich diet. So you can see there's a huge variation, just like us. That's the basis of doing GWAS in humans. People are very interested in trying to understand genetic differences, and as you've heard this many times, your genes are probably, how you chose your parents probably matters a lot in how long you're going to live. So we see this amazing diversity. So then we ask this other question, well, what happens when we do diet restriction on them? So we find this very striking effect. These guys, look at this line. This animal lives 4 times as long, right? This strain, I should say, not animal. So this is each fly, that we have 100 to 200 animals in each of them, right? Now, so these are doing well. You would say, oh, this is working great. Now you start looking here. These guys are already long-lived. Many of them don't do very well now on diet restriction. But it makes sense in some ways, right? If you've already got the genetic makeup, think about that TOR, which I talked about earlier. If TOR is turned down, then you're not going to have further benefits of diet restriction. So it may be getting the sweet spot. So looking into the future, I think this is going to be very important because like everybody can't take there's no one size fits all. And that's what the humbling knowledge, that's what knowledge is telling us. And this has been done for hyperglycemia. People have given different sources of carbs, like bananas, rice, and all that. And then they did this in 1,000 people. And then they asked, well, how does their glucose change? Well, guess what? It didn't change the same way. Maybe rice isn't bad for everybody. Maybe rice is bad for some people. So there's a lot of variety, and I'm sorry to sort of confuse you, but the problem is, yes, there isn't no simple pill or simple answer. There's going to be a lot of variations across people, but there are ways to think about this more creatively. Like, say for example, what I've learned from this is like, well, when you eat less, make sure you're not eating so little that your body is saying, well, this is really not good for me, right? So I think what's important, for example, we find is when you eat less, you become more physically active. So I think that's one way to learn. Maybe there's a sweet spot for you to eat this much, and as long as you can be more physically active, I think that's one way to kind of say, okay, that would be better for me than trying something that could be more drastic.
So we had a question about protein, and I had a question written down about protein. I read studies that suggest that as we get older, older adults need more protein. Is that the case? How should we approach protein?
So there are two parts. There was a very well done study on this question. What we had found before, by the way, protein is what turns on TOR. So unfortunately, when you turn on TOR, like I said, protein is also what's going to increase your risk of cancer and switch on TOR. But, like rightly said, as you get older, you may have more need for protein. So they did this study, and what they found, I'm just going to say what the study was, that under 65, being on a low protein diet was better for you, right? And one of the surprising things we have found from flies, it turns out it's true in humans and mice as well. These animals that are on the dietary restriction, these flies, they actually have more protein mass in their muscles, right? And the reason is they're active, right? Because it's the difference between being sedentary and being active. This behavioral switch we found is just as important. So I'm not going to show the study, but we did this cruel study where we cut the wings of the fly. And we found that if the animals are not active, they can't get the benefits of dietary restriction. So I think activity is really a part of it, because without activity, like I said, you can't burn your fat. Right? So you'll get some benefits of eating less, but it's got to be really, I think, like I said, I don't think there's going to be magic pills aren't going to be that easy. It's like really have to do all the hard work of being more active, sleeping at the right time, all the things your grandmother said you hated to hear. You have to do all that stuff really to ensure a longer lifespan.
So which fats are good, and why should we eat a low protein diet? Talk about fats, good fats, not so good fats.
Yeah, well, generally the trend from a lot of studies is that animal-derived fat and protein, those are sources to avoid. Avocados, for other plant-derived fats are good. But I like this advice that heard many times. Basically, try to eat whole foods rather than trying to extract things from food, because that's where we've created the imbalance. Again, remember, think about evolution, right? We grew up with whole foods around nuts and berries and whatever. Eat that. When we start extracting oils, we are creating an imbalance, and then we are asking the body to deal with it, right? So as much as you can, go to more whole foods. That's my simple advice, is what I say.
Okay.
I mean, I have to say, I just want to say, it might look like I know something about nutrition. Everybody I think has an opinion upon nutrition, and I realize that. And I do want to say, the more we learn about nutrition, I feel the less I know, because there's so much more. So I feel the least qualified to say anything, by the way. So even if I'm saying, I'm just telling you what few things we've understood, but it's a huge field and very complicated, and food is one of our favorite activities, eating, so everybody has an opinion about it. So yeah.
So somebody asked about alpha lipoic acid and about like dosages to take, and somebody else asked about, so what supplements do you take?
Private. Yeah. Well, it changes depending on what's happening in the lab. But sometimes Gordon says this is good, and then sometimes, well, no, I think this is working in our worms. So, but some of the ones that are out there, lipoic acid, nicotinamide, things to reduce cholesterol, these are some of the things that there are more studies on. Definitely vitamins are, multivitamins. There are certain vitamins that we do get deficient with age. Vitamin D, for example, Gordon's worked on a lot. So there are definitely some common sense things, and I do give into that, and I try to make sure that I'm not deficient in one thing or the other. I think the risk, the advantage of these supplements is there's less chance of doing harm. But I do think now I learned it's really a growing industry. It's a $21 billion industry now. And some cases we discuss this all the time with there's some things you can see way better effects with supplements than you can see with any of the known drugs. So why not take supplements? And I do think that there has to be, if the proper science is done on the supplements, and which there's a lot more coming, if you do proper clinical trials, there is a lot of room to understand this. Every time we find something, we go to, what's interesting is you go to people who do pharma, they go, no, we don't want to do anything with supplements. And there's this divide. And but on the other hand, I go to clinicians, they go, there's nothing wrong. If this works, we'll prescribe it. So I think there's a problem with the way, it's a bigger philosophical discussion I don't want to go into, but how society is organized. But I think it's more around how the patents are organized. Because companies can't make money on supplements, they've received a bad rap, I think. But I think it needs to be really, the science has to be done, and we should be doing that more proactively before we say they're not good.
So somebody asked about metabolism slowing as we age. So is that true? And is that part of why the good old middle-age spread happens? And is there anything that we can do to reverse that, to speed up our metabolism as we age?
Well, a very common sense advice, you all know. Again, I think eating less and exercising really helps deal with slowing down these changes. But yes, it does. There's a lot of evidence that it does slow down and changes. Like I said, you don't make as much ATP, and the best advice is what we just talked about. Yeah.
Great. So, how has your diet changed over the years since you were that first, young scientist that went, oh my gosh, dietary restriction? So talk about just kind of the evolution of how you eat.
Yeah, I have to give up all the things I like a lot. I had to give up Indian food, a lot, or cut down drastically. Yeah. Well, I think a lot of it is cultural, I have to say. I'm like, since my kids are, I have 2 daughters, and now it's different. I have to share ice cream with them, and which is something I think is not something I have to avoid. But I think what I try to do is everything in moderation, and I think because we are social beings as well, so there's other things we have to think about as well. But like I said, going towards a diet more towards whole foods. Less added sugar whenever we can avoid that, less meat-based products. Again, it's very easy to sit here and give this advice, very hard to practice. It's all about practice then preaching is very easy in this part. Yeah.
So, final question.
Eating at the right time, that's a big one, yeah.
Right.
Yeah.
So, Pankaj has 2 gorgeous young daughters. I can't tell you, they're the cutest little girls. So how do you talk to them about food? Because people here have kids, people have grandkids. Obviously, the best case scenario is to start off young and get good habits established. Is there anything how do you approach that with your kids?
What is amazing is, I have to say, because we've gone through through phases where my daughter wants to eat ice cream for breakfast as well, and for lunch and dinner, and then saying no, we can't have that. And what's very interesting is that we are creatures of habit, and we can be trained to do things. And so once it's taken out of the equation, she forgets about it, and there are other ways to deal with it. So I think definitely paying attention, to it. I don't eat soda, and we don't have soda in the house. Paying attention to things like that, just eliminating them. They don't know about it. They're fine. It's like when they see these things, like I said, we are just like any other animal. We gravitate towards this stuff, but even though it's not good for us. Yeah. So I think some way, once the rules are established and they understand, and I think I've been pleasantly surprised many times that she'll come and tell me what I'm supposed to eat or not.
That's great. All right. So we have reached the noon hour. It is time to wrap this up. First of all, many thanks to Pankaj for being here today. Many thanks to you for being here today. Pankaj is going to hang around a little bit if you have special questions. I want to wish all of you the most wonderful Thanksgiving. Enjoy your food. Eat well. Please talk to our staff people outside about becoming members, and thanks again for being here today.